to share with you some of my World Labs work peptide in tobacco. So I'm Yongfeng Guo from Tobacco Research Institute, Chinese Academy of Agricultural Sciences. So I'm going to tell you about several peptide families in tobacco, which are potentially involved in osmotic and salt stress responses. So as we know that hormones in animal systems are all or most of them, at least like 70% of them are peptides, including well-known or we are now familiar with, like this one is a diagram of how insulin, the animal peptide signal or hormone is produced. It is encoded by gene and cut or processed by protease and release several fragments of peptide, which are active in activating downstream signaling pathways. So in plants, we also have such kind of peptide signals, or we call them peptide hormones. Since the identification of the first plant peptide hormones, which is called histamine, from the early 90s last century, a lot of peptides has been identified in plant systems, in different plant species. In general, a peptide encoding gene is like the structure of peptide encoding gene is like this. There's a signal peptide at the N terminus, there's a variable domain in the middle, and there's a C-terminal domain, which is often conserved between among family members. So eventually, when after translation of this protein will be functioning to cleave this peptide from we call it proprotein to release a peptide, which is usually from 5 to 50 amino acid long, and often involve like a post-translational modification of this peptide to release to form a mature peptide, which can serve as a signal or as a ligand to receptors on the membranes to activate the signal inside the cell. So far there are like a lot of peptide genes or peptide hormones have been identified in plant. If you look at this picture, there's almost peptide involved every process of plant development and stress responses, including the mesostem, the vascular, the stomata development, and plant's response to various stresses, including drought, salt, and others. So if you look at the peptide already identified, which are involved in stress responses, you can see like some of the peptides have been reported to involved in responses to extreme temperatures. Some of them, like the CLE25 and others, are involved in drought response and salt response, and also some other types. And also, of course, a lot of peptides have been also identified in regulating plants' response to diseases like pathogens. So today I'm going to focus on peptide we have identified in tobacco, which we see we can predict, or we have tested them preliminarily to show they have a role in regulating plant stress responses. Before that, I'm going to show you some of the work we have done in different plant species in identifying peptide encoding genes. We use a pipeline or workflow. We set up like we based on like a size and the C-terminal conserved domain and the whole structure of the of the protein to predict the potential small secreted peptide, which are encoded by different genomes of different plant species. For example, we have identified like about 1,000 peptides encoding genes in tomato signal genome, and which is like numbers like around 5,000 in wheat. And we have like shown that peptides from from the same family in different plant species are very much conserved. If you look at this diagrams, there are like 21 CEP peptides in tobacco, 15 in Arabidopsis, and 13 in wheat. If you look at the amino acid in the peptide, you can see a lot of them are conserved, highly conserved, such that we can, I'll mention it later, a lot of time we can use a peptide from a different species to activate the similar signaling pathways in a different species. So one of the peptide family I'm going to tell you about today is called CEP. It's called C-terminal I forgot the full names, but it's just based on the C-terminal domain of this peptide. Generally, there's like around 15 amino acids in this peptide. If you look at the family members, we have like identified like 30 something peptide, CEP peptide encoding genes in the tobacco genome. If you can see the conservation of the amino acid, especially some of the like the glycine, the histidine, and the serine, and they are very highly conserved. Based on the sequence, as I told you earlier, these sequences are all located at the C-terminal part of the protein and was cleaved later on and serve as active signal molecular. So based on the structure of the peptide, this tobacco encoded CEP peptide can be divided into two types, type 1 and type 2. You can see the conserved the amino acid of these two types of CEP peptide. Each in one of the peptide, if they are in the same type, they are more highly conserved. So next we checked if these peptide encoding genes are induced by different or can be induced by different stress response, stress treatment. So we checked the promoter region of this genes and find there are quite some like stress responsive cis element on the promoter region, and also of course hormone responsible regions too. So we went on to check the expression of some of the peptide encoding gene, and we found that quite some CEP peptide encoding genes can be operated by different stress treatment, including like high temperature, low temperature, osmotic treatment, and stress treatment, and salinity treatment, salt treatment. In this case, sodium chloride. You can see quite some of them can be induced by one or more different treatment. So we picked some of the peptide that potentially are upregulated or induced by by different stress treatment. So we sensitize the peptide. The good thing about peptide studies, you can sensitize them chemically to use them to treat, in this case, detached leaves, to see if they can play a role in stress response of plants. So in this case, we picked some of the highly induced genes in response to sodium chloride to see if they can play a role or change the plants, or in this case, the leaves response to treatment. So you can see this is on top 2 rows are water treatment. After 4 days, essentially there's not much change, especially for the white type leaves. But when you have a 150 molar of millimolar of sodium chloride, you can see yellowing of these leaves under salt treatment. And some of the peptide, if applied in the solution together with the salt solution, then can elevate the yellowing process of the leaves triggered by the salt treatment. These are chlorophyll compound after treatment. You can see there's at least like CP5, 17, 14, and 13 can have showed like more greener leaves after treatment in comparison with the treatments with salt alone. And of course, we checked another one more experiment with CEP17, which is germination on plate after, of course, after salt treatment. We can see that the CEP17 peptide treatment can also increase the germination rate after salt treatment and Arabidopsis, but in this case, it's tobacco seeds. So this is of course, another experiment we did is tried osmotic tolerant treatment. So similarly, we use Manitou to mimic the osmotic stress treatment. So like after 5 days treatment, you see the yellowing of the leaf discs in the control, but you see greener leaves in the 4 peptides from the 4 different types. This is similar in the chlorophyll content changes. We also did a Manitou treatment and seedlings, and of course, this is a Manitou treatment of seed germination. Again, we see that this is the Manitou treatment will inhibit root growth. You can see CEP17 peptide will increase root growth, even together with the Manitou treatment, but not without Manitou. This is very interesting. So with only medium, different peptides are different, but after the osmotic treatment, some of them can increase, but some of them can decrease plants to the tolerance to the osmotic treatment. This is a seed germination experiment showing that CEP17 can also, the peptide can also elevate the plant's response to osmotic stress in seed germination. So that's all I want to tell you about the CEP family. Now I switch to another family, which is called the CLE family. We found 41 CLE family members on the tobacco genome. So this is the phylogenetic tree together with Arabidopsis, and I think that this is tomato. So they group in different groups, and each group will have a very highly conserved amino acid of the peptide region. So we did also did the analysis of gene expression change after, of course, in this case, we did expression change after, I think this is salt treatment. Yes, salt treatment. Some of them are increased expression, like CLE12 and CLE29, but some of them has the opposite change in expression. So some of them are activated, some of are inhibited in terms of transcription. Sorry, I think that's osmotic treatment, the changes of expression. So we did a similar experiment with osmotic change using synthesized peptide to see if there's a change in response to osmotic changes with using mannitol. So this is CLE29. So you see this is alone without treatment. This is just detached leaf. This is detached leaf treated with mannitol, 300 millimolar. And if you treat together with the peptide, you can see a little bit greener leaves in comparison with mannitol treatment alone. So it's the same for CLE12 too. This is more obvious if you treat with the discs, leaf discs of CLE12 peptide together with the mannitol, you will see slow down of leaf yellowing in this case. So this is a salt treatment. We can see some of those peptide encoding genes are increased during salt treatment, like 12 again and 41. So we did the similar experiment with salt stress treatment and peptide treatment on leaf discs. You can see that this is 150 millimolar sodium chloride. After 4 days treatment, it's obvious yellowing compared with the control treatment. But if you treat together with peptide, synthesized peptide, you will see elevation of this yellowing process in response to salt treatment. Similar, we have similar phenomena from CLE4 peptide too. So that's something very interesting. We see peptides from two families, CEP family and CLE families. There's always some of the peptides can be used in reduce the sensitivity of plants to salt or osmotic treatment. So now I'm going to show you data from another family. It's called the PSY family. This family is a little bit different. So we have 9 members of PSY families genes in the tobacco genome. And also, of course, from analysis of the promoter regions, some of them can be potentially induced by stress treatment. And indeed, when we treated the plants with different stresses, this including mannitol, salt, low and high temperatures, we can see almost every of peptide of this family can be induced by one or more signals. So we did the treatment as I showed earlier, but surprisingly, these peptides, we don't see any increased tolerance after the treatment. We only see increased sensitivity after treatment. A lot of them have, of course, Yeah, most of them will have an increased sensitivity, or only one of them showed increased tolerance, like this is PS1 1 here. Like this one too, to this previously to salt stress, this is to mannitol or to osmotic stress. You can see PS1 and PS3 all in comparison with control after 8 days treatment, you see more severe yellowing phenotype of the leaf discs. So we also tried some other like including whole seedlings and seed germination. So all show that the PSY peptides, a lot of them, most of them, will have an increased sensitivity after treatment, together with salt or mannitol treatment, indicating that different peptides from different families will have different roles in terms of in regulating plant response to stress treatments. We chose some of the peptide with from the screening to treat whole plant in part experiment and in the field. This is just some experiment to show you we can treat one of the plant, tobacco plants, with one of the peptide, and this treatment can increase significantly the tolerance of the plants to drought stress. This is drought stress after 40 days drought treatment and after rehydration of the plants. You can see the significant difference of the phenotypes. So this is tomato plants, also drought stress treatment. Two of the peptides can increase the tolerance of to drought. So this is wheat, also increased drought tolerance. So as I mentioned, these are not the peptide from one. You don't have to use a peptide from the wheat to get this kind of response. You can use a peptide from tobacco or Arabidopsis to have similar responses once it works in the original species. So this is a peptide treatment to increase wheat plants' tolerance to salt treatment. You can see this is the treatment. This is control. Oh, this is another. This is a cauliflower head after harvest. When we treat this cauliflower head with peptide, we can increase the shelf life or keep the cauliflower head fresh for a couple of more days. So this is the tobacco plant in the field. After treatment with peptide, we can see significantly improved growth and development of the tobacco plants.